Tim, thank you for a very kind introduction, and Maria and Tim for the invitation to be part of this fantastic event and a pretty prestigious group to be involved with. Helen Marshall, Dan Thomas, Shilpa. I'm the bunny in the middle, but hopefully I will give you one or two pearls of wisdom about my journey. And my approach to this is going to be quite personal. I'm going to talk you through some of the bumps that I feel that I took along the way. So I've got a disclosure, and that is when I started my path in medicine, it was with one purpose, and that was to be a clinical doctor who taught individual patients. And that was the path I wanted to take because I had a wonderful role model, my father. My father, Greek-Australian, one of the first Greek-Australian general practitioners in this city, worked for 50 years as a solo GP, 8 in the morning, often coming home 9 or 10 at night, and I never heard him complain once. And I thought, that's who I want to be like. So I had a very linear path through medicine. High school, undergraduate med, Uni of Adelaide, 6 years. Met Dan Thomas just before we started in 1993. Internship, basic physician training, and along the way I decided I wanted to be a cardiologist, and I did my 3 years of cardiology. Research did not even come into my thinking. Infamously, famously, when I was interviewed for my position in cardiology, I was asked if I wanted to do research, and I'm a very honest person. I replied, I might do the odd audit here or there. That was my plan. Well, having said that, I am an inquisitive person by nature. I don't tend to take things as they are on the surface, and I wanted to ask about the why and the how. Why was I seeing so many people, despite all of our treatments, still dying of heart disease, and how was I going to fix it? And in the 3rd year of my training, I decided to embark on a PhD. Now, I approached a clinical mentor. His name is Stephen Worthley, and Steve was a very energetic cardiologist who'd just come back from New York, and he was establishing a program in cardiac magnetic resonance. And Steve offered me a PhD to use MRI to image the hearts of people with diabetic coronary artery disease, diabetes obviously being a very important risk factor of atherosclerotic heart disease. I thought, what a beautiful project. It felt safe. I'm going to sail through this, no problems. Well, timing is everything. And the same night as I had that first meeting with Steve, I sat next to a very iconic professor of cardiology here in Adelaide, and some of you would have heard of, John Horowitz, at a cardiovascular registrar's dinner. And John asked me what I was going to do, and I said, I'm going to do a project on cardiac MRI, John, and diabetes. And John, in his provocative way, retorted, he said, Peter, there are 2 types of research projects that students take when they do a PhD. Some of them, they know the answer to the question pretty much as soon as they ask the question, and others will lead you down curious new paths and you will find something new. You're about to do a project where you know the answer before you embark on the mission. Now, whether John was right or wrong is for another day, but that really weighed very, very heavily on me. And I took the longest ever shower I've ever had the following morning, and I decided this MRI project is not for me. I want to do something new. And so I sat down with Steve, politely declined his offer and said, Steve, maybe not that one. I'd like to do something cutting edge. How about stem cells? Now, I knew nothing about stem cells, absolutely nothing, and I did do the first 2 or 3 years of basic science in medicine. I knew nothing about it, but I knew that stem cells were going to be a thing of the future for mending broken hearts. Steve could have done something very easy. He could have directed me to Richard Harvey's lab in Sydney or another lab in Melbourne or maybe overseas, but he said, you know what, I know a stem cell biologist here in Adelaide, and he's got a friend, and they were Andrew Zanatino and Stan Gronfos, the Batman and Robin of mesenchymal stem cell research in this country. And that was the best introduction I ever had, except to my wife, actually. Andrew and Stan adopted me into their basic science lab. It was a fantastic lab environment. They knew nothing about hearts. We knew nothing about basic science or stem cells, but it was a marriage that was built around support and positive, optimistic, hopeful working together. I can tell you in the first year of my PhD, trying to work out how stem cells were going to fix broken hearts, I felt like a complete dud. I used to stand over my fume hood watching these other much younger basic scientists. I was 30 at the time. I was watching these other basic scientists go about their day-to-day work knowing exactly what they were going to do. I couldn't hold a pipette. I didn't know whether the test tube went up or down. I had never felt so vulnerable in my working life. I'd never relied on anyone before. I knew what to do in clinical medicine. I did not know what to do in basic science. That was a very humbling experience, but I also learnt the power of asking people for help. Because, as clinicians, basic scientists find us a little bit of a novelty in the lab, and they're only too pleased to teach me what they knew. In addition to doing a lot of in vitro cell biology work, I performed the first ever study of a technique of stem cell injection across the lining of the heart using a very sophisticated mapping technology. This is 2006. I was the first person in the world to inject stem cells using this technology into a model of heart failure called dilated cardiomyopathy. And I trained overseas for about 3 weeks to learn the technology, brought it back to Adelaide, and there I am working on sheep in what was the old IMVS. I still got to do MRIs because we used to MRI these sheep under the cloak of darkness every weeknight at the old Wakefield Hospital because we didn't have an MRI scanner at the Royal Adelaide. Now, I didn't publish any cell science or nature papers, but with hard work and with Stan and Andrew's mentorship and support, we got a number of first author publications. So what did I learn from my PhD? Well, first of all, I learnt that if you're driven and you're self-motivated and you've got good mentorship, it goes a long way. We didn't have an established stem cell cardiovascular program. We took a risk. I took a risk going into basic science. Those guys took a risk in getting into cardiovascular regenerative research. We worked hard. There were a lot of ups and downs and a lot of self-doubts. I may or may not have cried at the beginning of my 3rd year in my supervisor's office, but we made progress and we established something. I learnt that working smart is as, if not more important than working hard. My first year, I made some progress. My second year, I thought I'm really ready to ramp it up. And I had been used as a medical student to working 16-hour days, studying, and then as a clinician, I thought I can do this in a basic lab. Well, coming into work at 3 or 4 in the morning, trying to do 5 or 6 experiments before 8 a.m. when I did my sheep surgeries, and then MRIing till quite late at night, used to give me stomach ulcers. And I found that doing that day after day after day did not translate into results. Yes, I was multitasking. I thought I was being smart, but I was being ineffective. And this was probably the most important first lesson I learned about research. It's working efficiently. I learned that research progress does not happen overnight. As a clinician, I was rather impatient. Research doesn't always come out with linear outcomes. You have to be patient. And I learnt, and this is one of a colleague of mine, Steve Nichols' favourite sayings, it takes a village to get it done. I relied on much younger people, scientists, to show me the way. When I was doing those sheep surgeries, I needed someone holding me a cup of coffee with a straw so that I could drink from it, or to move the image intensifier. I needed help, and I learnt not to be afraid to ask. And I learnt that there was no substitute for great supervision and mentorship, and everything I learnt about mentorship began with Stan and Andrew. Now, at the end of 3.5 years, one of my clinical colleagues said to me, "You've got your basic science out of your system. Now go and do your clinical fellowship, start making your money, and you can come back and you can lead basic scientists doing research in your own lab." My response to that was, I'm not ready yet. I actually know nothing about basic science. I need to go and do a postdoc. And in my field of cardiology, it's a little unusual to do 3.5 years of basic science, let alone extend that further. I decided to go to the Mayo Clinic, and there are several reasons behind that decision, but primarily I wanted to work with a man called Robert SAHMRI, who had just been announced as the American chair of the Stem Cell Cardiovascular Network. And I thought, if I go to Mayo, having worked with these stem cells in test tubes and sheep and pigs, I'm going to start putting them into humans. Unbeknownst to me, they weren't doing any of these stem cell trials at Mayo. Rob was simply chairing the National Committee. Rob had a very small, non-NIH funded basic science lab that was focused on vascular biology, atherosclerosis research. And my first meeting with Rob, he asked me a question that has plagued me and challenged me and excited me for the last 10 years or so. He asked me the question of where hematopoiesis first begins in developing embryos. I had no idea. He replied, it begins in the aorta. If it begins in the aorta, I'm going to challenge you, Peter, to find hematopoietic stem cells in the aorta after birth, because I think these are the stem cells that cause blood cells, immune cells, to form in developing atherosclerotic plaques. The paradigm was that these cells came from the blood and from the bone marrow. I'm going to challenge you to show me that they come from the vessel, the artery wall. Well, Rob sent me on a little bit of a wild goose chase for a while there. I was doing mouse work, having moved from large animals to mice. My dad visited me at Mayo. I took him in on a Saturday night to feed the mice, and he said, I trained my son to be a mouse doctor, and he was right. But it was new, and I was learning. I also joined another mentor, Martin Rodriguez-Porcel, who was an up-and-coming clinician researcher who was interested in molecular imaging of stem cells, and Martin got me working in rats, so I had 2 small animal models at once. I took my then 3 children, I now have 4 children, and my wife with me for just over 3 years, and we braved the minus 25-degree winters of Minnesota that lasted 6 months, and I was in a small laboratory, not particularly well funded, but we had a great time, including growing moustaches. Now, Rob wanted me to find broad-acting hematopoietic stem cells just like there were in the developing embryo, aorta and in bone marrow. After many, many experiments, it didn't quite map out the way we expected. What we realised we were seeing was a very specific type of stem or progenitor cell that was producing not all types of blood cells, but one type, and that was the macrophage. And that led to some studies that were published at a time where some really interesting things were happening in macrophage biology, including how they might be formed in tissues. And we described what was really the first ever macrophage stem cell in the outer lining of arteries. So what did I learn from my postdoc other than the science? Well, I learned you need to do the time. I see clinicians and medical students who are on a pathway like I was, where they want to get out to the other end ASAP. My cardiology registrars think that they need to be consultants by 33 years of age. Well, I can tell you that we end up doing what we do for a very, very, very long time. And in all of my years of working, my best years were in my 30s when I was doing a PhD and a postdoc. I was working hard, but I was smelling the roses. I was having experiences here in Adelaide and overseas. Life is not a race. Don't look over your shoulder at what other people are doing and what they're achieving. Tread your own path. Steve asked earlier about going overseas. We were apprehensive about going overseas. We're a Greek-Australian family that love being around our parents and our brothers and sisters and cousins. Our 3 years in Minnesota were the best years of our life. We met people from all over the world. We visited the Dakotas, we visited Iowa, we went to Wisconsin. We did all the sorts of things we would not have done here, and I worked in a great environment. I learnt scientifically not to ignore unexpected results. Yes, you form a hypothesis, but when that hypothesis doesn't work out, if your research was done well, pay attention to your data. The most interesting projects, in my experience, are the ones that led me into new avenues. And Dan mentioned it. My postdoc supervisor's mantra is, we are truth seekers. Science is about finding the truth, and you must not ignore it. And I found out once again that you've got to enjoy what you do. Now, I spent 3.5 years at Mayo. I loved it. I was asked to stay on and do some clinical and scientific work there. But my 3 children knew the 50 states and capitals of the United States, and they couldn't even remember where their hometown in Australia was. So we had to get back. And I decided I'd come back via Melbourne, and I spent 2 years after a 7-year hiatus from the clinic learning how to put stents in people's arteries, and that was terrifying. I could do microsurgery in mice, but putting stents in, doing angiograms and stenting human patients after all of that time away was probably as intimidated as started basic science in the first place. What did I learn from that? It's just like riding a bike. You pick it up very quickly, so don't be afraid of taking a break from the clinic, going to basic science and then coming back again. It's not that hard. 2 years away from basic science doing clinical work, got my juices flowing doing clinical research, and that's rounded out my research training very well. And then in 2015, had some offers. Do I go to Sydney? Do I go to Melbourne? Do I go back and run a lab at Mayo? SAHMRI came knocking along with the new Royal Adelaide Hospital, and an opportunity, perhaps most importantly for me, to return to my family after what was just over 5 years. And to be truthful, I'd love to say I came back, Steve, for this building, but for us it was time to be reunited with our loved ones, and I'm grateful that we did. It helped that we were coming into a fantastic institute, and especially for me that I was coming back to work with people I really wanted to work with. We've heard the power of partnership and collaboration. Well, for me, coming back to work with a long-standing friend and an inspiration in Steve Nichols, who at that time was head of our theme in heart health in SAHMRI, was fantastic. Steve was a gun in clinical trials and coronary disease research. I was bringing the basic science, some more interventional cardiology, and for a number of years we formed a good partnership. We added to the family. 2 years later, we got a fantastic basic vascular biologist from Sydney, Chris Bursell, who now co-directs our group with me, and we form a bit of a yin and a yang. We complement each other's strengths and weaknesses. And along the way, I met a biochemist and molecular biologist, Chris Proud, who has taught me an enormous amount, not just about rounding out my research skills, but how to do it with dignity, integrity, and real rigour. And I still value Chris's collaborative relationship enormously. Now, this is a photo of my group a couple of years ago. I'm sorry that I don't have a more recent one. What I wanted to highlight here is that I'm a clinician doing basic science, and every person but one in that photo is actually a basic scientist. So if you're a clinician and you want to join my group, please come and join. And great to see Richard Lee, who's recently joined us as a Flinders student. We need clinicians doing basic science in our lab. The sorts of things that we investigate are fun and exciting, and we need people to bring clinical acumen and perspective to help answer important questions. So briefly, what do we study? We study the disease that underpins the leading causes of death in Australia, atherosclerosis, the plaques that grow in people's arteries. We particularly are interested in the interface between inflammation, particularly innate immune cells like macrophages, monocytes, neutrophils, and the way they interact with new blood vessel formation in those atherosclerotic plaques. Our models take us into coronary artery disease, which is my clinical bread and butter. We're working with vascular surgeons in the area of peripheral artery disease and recently with stroke physicians in stroke. And along the way, we've developed a real interest in wound healing and also in cancer, particularly in melanoma. How do we do it? Well, 9 years of research and then clinical training has to be good for something. My program allows me, through my training, to do really what is a discovery all the way through to clinical practice and implementation platform. We use lots of different models, lots of cell and molecular biology, mouse experiments, and access, importantly, to human biobanks, blood and tissue, registries, trials, and we have Australia, perhaps the world's leading core laboratory in coronary imaging, which underpins some of the clinical trials we do. This is not to brag, but it's to show you that I'm involved in lots of different areas of research. We have a stem cell program. We look for novel targets in heart disease, including novel kinases. We look at repurposing old drugs like colchicine, and we have a particular interest in diabetes and its interaction with heart and vascular disease. I alluded to my postdoc discovery. I just wanted to show you that 3 years at Mayo ultimately amounted to something. Those stem cells that I discovered in the wall of arteries ended up not just producing macrophages, but they are a bipotent stem cell that also forms new blood vessels and endothelium. Scientists had postulated for 100 years that in postnatal tissues there would be a stem cell called a hemangioblast, and we think we are very close to demonstrating that this stem cell is that hemangioblast. And we've taken our work from the mouse aorta to study it in muscle and muscle repair, skin and wound healing and melanoma, the heart and myocardial infarct repair, and recently the lung and adipose. And with some early clinical forays, we're now finding these stem cells in human tissues, which is exciting. As I mentioned, we do a lot of work as to what causes atherosclerosis and how to treat it. And one of the exciting platforms is to take an ancient drug, which is colchicine, a drug used to treat gout, and to actually use it to block the inflammation in plaques. And we have active work in the mouse space and in the cellular space, looking at how this works. And in a month, we'll be finishing a clinical trial using our plaque imaging technology, OCT, like Justine showed us earlier, to see how this drug works in coronary plaque. Collaborations are everything. SAHMRI is a wonderful institute because we have people doing different things just down the corridor. And one of our mantras is to break down those silos of research and to reach out to each other to find out what we're doing. I spent years having tea room conversations with Tim Sargent over there on the right of screen. Tim is an autophagy researcher who's interested in how autophagy causes dementia. Well, it happens that the plaques in dementing brains are very similar in some ways to the plaques in heart arteries. And Tim and I, with Julian Ben-Salam, are now using Tim's blood test for autophagy to see how it might be a new biomarker for patients with coronary and peripheral artery disease. My best collaborations are with people actually outside of this building, and in particular with engineers and physicists, because they think very differently, very, very differently to me. Mergen Gaius is a mechanical engineer who is a gun in modelling mechanical shear stress in new ways. I bring the expertise about how coronary plaques develop, form and destabilise. I bring the imaging technique, and Mergen and our PhD student Harry model both in silico and now using human imaging. They model how plaque progresses because of shear stress. And not too far from Mergen, I work with Rob McLaughlin and Jarwen Lee. So Chris Bursell and I have a number of grants and papers that we share with these two physics-based engineers who are interested in developing new light catheters, hybrid catheters, that take the sophistication in how we image plaques to a completely new level, so that we're not just looking at the anatomical construct of what's in the plaque, but the active dynamic biology. And we've taken a really exciting project from mouse models in plaque. We're just starting our first pig model, and we're now working with human specimens as well. So how do I do it? Well, I have a busy and varied life. And this is, again, just not to brag, but it's to show you in colour coding how I divide my time. I do a lot of clinical work, so I have to be out of bed at 5:30 most days. And most of the time I'll have patients in private hospitals, and I do angiogram lists at 7 a.m. on a Monday morning before I come to SAHMRI by about 9 for my meetings. So that's at the edges of the day, my private cardiology. Monday is an very admin-heavy day, lots of meetings about how we're going to make SAHMRI and the lifelong health theme better. Tuesday, I spend my entire day doing angiograms and stents. Wednesday is my day of consulting. That's why I wanted to be a doctor in the first place, and I still love consulting. I still love interacting with individual patients and having that immediate satisfaction of helping them get better. Wednesday night, well, I get punished Wednesday night because I'm on call for 24 hours covering the Royal Adelaide and the Queen Elizabeth to wake up to treat heart attacks. And the older I get, the harder it is to wake up and the harder it is to back up the next day. And Thursday and Friday are my fun days, because they're the days I really do my research, supervising and mentoring my students and meeting with my collaborators. A wise man once said to me when I started research, you do not produce in research if your motto is that I'm working 8 to 5. Work does not get done if you're not prepared to push the boundaries of evenings and weekends. And almost every weekend will involve me opening my laptop and doing some form of work. So, what are my ultimate lessons as a clinician scientist? Diversity and balance is what keeps me energised, energetic, and what allows me to be energetic when I'm a clinician and energetic when I'm a researcher. We do what we do for a long time. Diversity is what makes it, for me, worthwhile and enjoyable. Collaborations, especially the ones that take you outside of your paradigms and thinking models, are what help me generate new research ideas. They bolster my weaknesses and they fortify my strengths. Research satisfies my intellectual needs and my creative juices. I'm much more stimulated having meetings about research respectfully than putting in a stent in the middle of the night. But the wins in research take a long time and they're sometimes not predictable, so be patient. Supervising my students, both basic science and clinical, is the highlight of my week. But if you're going to supervise, commit to it properly. I've told you about my good supervisors. I've had some shoddy ones along the way too. Do your students the justice of supervising them properly. Be invested in their work. For me, I could not drop clinical practice. So for those of you who want to maintain the life as a clinician and do research, it is possible. You're just going to have to work around the edges of the day and get the balance right. Being a clinician makes me a better researcher. I think being a researcher makes me a better clinician. And the best part about my job is I never stop learning. And that's what excites me about the next 20, 30 years. Thank you.